Good afternoon, Professor.
Good afternoon.
Students,
Yeah.
students, we'll start our second session for the day. We have with us now Professor Annamma Anil Modaner. She is from the DBT ICT Center of Energy Bioscience, Institute of Chemical Technology, Mumbai, a renowned industrial microbiologist. Her area of work is extractive biotransformation, design and engineering of enzymes, selective isolation and capture of natural bioactive molecules, secondary agriculture and its products, process integration and intensification, process development, characterization and scale-up. Professor Annamma has did a BSc in microbiology and MSc life science at the University of Mumbai. Followed up, she did a PG diploma in bioinformatics, and after qualifying the CSA JRF fellowship and ICMR JRF exams, she entered the ICT for PhD in applied chemistry and has worked there since then. Professor Annamma has authored and authored more than 77 research publications in various national and international journals, reputed journals. She has 39 patents, of which 37 is international and 2 is from national level. She is also a part of a team led by Professor Aravind Ralli that conceptualized the implementation of India's first ever 2G ethanol plant. It was inaugurated in the town of Kashipur in Uttarakhand in April 2016 and is capable of processing 10 tons of agricultural waste in a single day. So with this short introduction, I invite Professor Annamma to deliver the talk. The floor is yours, Professor.
Thanks for your very kind information, kind introduction, and this opportunity to present. I'm sincerely sorry because earlier I had to cancel the lecture earlier. So, right, we get on to the topic, and what I plan to take you through is in this specific presentation is to understand the critical nature of how we design medium where we allow the organisms to grow. Because when we are basing our entire wide biotechnology, that is industrial biotechnology area, we are looking at microorganisms as the major sources for making goods or manufacturing goods for the industry that we are needed, that are our needs. So for the transition from the petroleum-based industry to a bio-based economy, we basically need biological systems to perform as per our requirement for giving us the different products that we are intending to use in different applications. So any of the living cells, you can have plant molds. Am I audible to all? Is my screen visible and clear?
Yes, Professor.
Yeah, thank you. So basically, if we have to produce chemicals, plastics, pharmaceuticals, foods, energy carriers, multiple of these things, we need to look at living cells for their options. And therefore, the technology basically needs to use molds, bacteria, plants, and yeast to synthesize very easily degradable products that have applications in our day-to-day life. But when we are doing this, we also need to take into attention that we need to produce less, we should need less energy and produce fewer byproducts during this entire process so that the wastage is less, right? So when we look at, I draw examples from a kitchen of a house. Yes, the kitchen is the center area where we are making food for the entire people in the house. But if it is mismanaged and does not suit the requirements of the family or requirements of the people staying there, then it is an utter failure from that point. The food starts to accumulate. There is a lot of food wastage that happens. People are not nourished properly. So the housemaker actually needs to pay attention to the needs of each and every individual. There may be older people, there may be younger people, there may be kids, there may be middle teenagers. Each and every individual's requirement is different. Therefore, we need to optimize what we need or making in the kitchen so that everybody's requirements, needs are fulfilled. There is less energy wasted, and there are fewer things that are stored back after the food is being consumed. On a similar, taking analogy from there, on a similar lines, when we design experiments or when we design the entire platform to make industrial microbiology or industrial biotechnology successful, we need to design things or we need to optimize certain parameters, growing conditions, certain media conditions in a manner that we use less energy and we get fewer byproducts, allowing us to have a very good efficiency for our product. So white biotechnology today is being used for metabolite production, waste treatment, production of biocontrol agents, bio-based fuels and energy. So the applications are all over the place. And the industry is poised to jump almost twice by 2025 because there are a lot of things that are happening in this current sector. So what we are at the moment trying to do is understand how these different uh, areas where biotechnology is being used, and each of these areas has a different cost requirement. By cost requirement, I mean that if I'm looking at the health and medicine segment, drug production, personalized medicine, pharmaceutical proteins. These are molecules that need very high purity, and the agencies that are controlling these chemicals are going to be very stringent about how you are preparing the final product. On the contrary, if I'm looking at environment, that is the environmental biosensors, treatment of waste, bioremediation, pollution control, biodegradation. This is not an area where people will like to spend money. This is an area where, yes, they need microorganisms for their bulk treatment of waste, but you will not have a value, very high value, for the starting material or the microorganisms that you are providing in this. And therefore, stringent conditions, if you put for the growth of the microorganism, it will be definitely not appreciated in this zone. Okay. On similar lines, when we are looking at food or vitamin production, fuel production, bioconversion of natural products.
Professor, sorry to interrupt.
Yeah.
Are you sharing your screen? Because it is not,
Yes.
your slides are not visible.
I shared my screen right away in the beginning. No?
No.
Hold on. Can you see it now?
Yes, Professor.
Yeah. Yes, is it visible?
Yes, Professor. Yes, you can.
So if we are looking at the microbe area and the food feed segment, it may not be as stringent as a health segment, but definitely this too has its own requirements, and we need to have a very uh, streamlined knowledge of what is going into the production area so that the final product can be clearly labeled. Applications in the animal industry and the plant industry also share similar stringency when it comes to streamlining what is being used in the complete process. And therefore, when we look back at what are the steps in white biotechnology or taking any organism right up to the product stage, it involves a whole lot of processes or a whole lot of unit steps to actually come and see whether the product can be formed or not. So first and foremost, we need the organism. Where does the organism come from? Then if the organism is selected, Does it produce what is needed? How much of material it produces? Can we maintain the culture for a longer period of time? And every time we use the culture, will it give us the similar yields? Mass cultivation in bioreactors at a low cost, and finally, product recovery. And then, after product recovery, also, how do you discard this material or how do you reuse this material? How is the waste that is generated processed? So, all these steps are important areas that we have to pay attention to when we are developing a process in under white biotechnology. But today, what we are going to focus on is how do we develop robust microorganisms that have been isolated from the nature and take them through a route which is a profitable route for the final product formation. So the first part that we look at is isolation of organisms from the environment. Now, why microbes are environment important? So if you look at any of the cycles of nature, carbon cycle, phosphorus cycle, nitrogen cycle, you will always see that microorganisms form a major part or an integral part of this process. See, because it can convert the complex materials into simpler forms and make them available in the cycle again. And therefore, microbes have been given the right, have been given the position that they are driving the chemistry of life by decomposition, by diseases, multiple of these things. They ensure that the complex materials or the complex living forms of life are degraded and go back to the soil, and then from the soil they come back in their different forms. So whichever life form you see, be it human, animal, plant, fungi, any of the life forms you see, all of these life forms are going to be digested and decomposed by the microorganisms, allowing the molecules to be, allowing the complex polymers of the body to be dissolved and allowed to be decomposed completely to go back into the nature. And to, uh, to allow this process, we also have a whole lot of diseases and multiple other things that microbes actually cause. And therefore, from learning microbiology over the years, we have come up with applications and uses of this entire chemistry to make profitable processes out of. So we look at secondary metabolites, we look at bioremediation, we are looking at soil structure improvement, plant growth promotion, nutrient recycle, humus formation, multiple of these things. So microorganisms may be tiny, but they definitely are an important source. And wherever on this earth that you search, you will have microorganisms inherent to those zones. So each zone has its own population of microorganisms that thrive in these zones. And when we come to isolation, we need to look at which zone you're looking at and then identify specific properties of that organism so that you can enrich it and grow it properly. So when we are using microorganisms in technology, we would want systems which can use, or we would want microbes which can use a wide range of substrates. You would want microbes which can have biomolecules that can be produced. It should have the potential to be grown in large amounts. Bioremediation and decomposition may also be factors that we need to look at, because after the microorganism is grown in a large scale, we should be able to degrade it and decompose it so that it doesn't create havoc in the ecosystem. It should be available to modifications that we need to do. And to aid all of these, a fast growing organism is definitely preferred over other slow growers that you have. So using microorganisms for economically valuable products is our main focus of affair here. So how do you pick up organisms? So this is a collective slide I have made from different snapshots of different pictures from different publications. So when we are screening for different fungi or we are screening for microorganisms which have specific properties, we go through different stages. We collect microorganisms from the environment, and once we are done with this collection, we would look at how we can pre-culture them, then how we can subculture them, and finally, once we have subcultured, how we can store them and identify the different properties that it has. So this journey from collection to storage and identification needs different mindsets at different points. Mindsets meaning that when we are collecting organisms from a local environment, we have to bear in mind that that environment may not be the most best environment where the organism is thriving. The organism may be in its dormant state, or the organism may be suppressed by other fungi or other microorganisms that are living in its vicinity. So, we need to do is, with our final objective in mind, we need to be able to pick up this entity and allow it to grow in a manner so that you have final product. So, I mean, the analogy here is that multiple people appear for these music competitions, right? You have almost like lakhs and lakhs of entries. We had Indian Idol and multiple other music based platforms where they do a selection criteria. Okay. So, first and foremost, when you fill up a form, almost you have lakhs of people coming in. And from that lakhs of people, there is a pre-selection which happens, which sorts out all the non-performers and picks up the ones who have potential. And once you have picked up the ones who have potential, then they actually send them to maybe grooming classes or what do you say, training classes, which groom them further up. So you are doing an enrichment of their potential. And once you have done an enrichment of their potential, then you put them on platforms and give them different criteria of selection. You ask them to sing for Western songs, ask them to sing Hindi classical, Southern classical, and then you pick up the ones who can perform under different criteria. On a similar note, here in fungal systems, or when we are looking for microorganisms that perform better, the selection criteria at each level is going to be different. And therefore, the media that you use or the medium that you use for allowing the growth of these microorganisms is also going to be different at each level. And we need to pay attention to what are their requirements so that we can improvise on each of these and grow the culture in a systematic manner to allow us to be able to finally select the ones that we want. Many a times, these selection or screening procedures end up with bacillus species or serratia species, which are commonly found in the air around us, environment around us. Even if your labs and places are very clean, serratia, micrococcus, bacilli, staphylococci, these are commonly present in the environment around us. And most of the outputs of any screening experiment are these common organisms. So are we selecting them from the environment that we are picking up or from the niche that we are picking up, or are they part of the common air around you? Is a question that you need to answer. And therefore, your selection criteria or their selection parameters for the media that you are giving has to include some properties wherein that only those specific organisms from that environment will be picked up for further applications. So we look at media for selection and isolation. So when we are asking, when we talk about media for selection and isolation, we should ask ourselves, what is it that the microorganisms need? So you have picked up soil, or you have picked up a natural environment. You have picked up a decaying root or a decaying plant material, and you are expecting that all microorganisms in that collection we should be able to grow them up in a manner so that we can isolate them further. So the questions we need to ask here is, what is it that those microorganisms need? What is the source of energy? Do we need to give them special environmental conditions to grow and reproduce? What are the nutrients it needs? Does it need vitamins? Does it need growth hormones? What kind of material? Now, even before we ask this question, we should ask ourselves, what is the final output that you want from this screening exercise? You have to have a targeted output, because the intermediate steps are going to be in a manner that screens you towards a targeted output. Because what you are essentially doing is, we are at the top of a funnel and we have samples which have lots of microorganisms. And this entire process, we want to funnel ourselves and we want to come to one or two strains which are best performers. We are not interested in this whole category. There is some specific property that I'm looking at, and therefore I will keep screening or I will design my media in a manner till I come down to this specific one or two organisms which have the properties that I or my experimental plan requires. Okay. Once you have defined this aspect of it, then the selection of culture media to reach towards your final goal becomes very easy. What in this decision making, what we also need to think is that in the natural environment, the microorganisms have adapted to whatever you give them. And therefore, your conditions in the culture media that you are forcing these microorganisms to grow must satisfy all these requirements. And that essentially becomes the different types of media that you are. Okay. So, parameter that you need to keep in mind is, there are certain nutritional requirements for growth and normal functioning in every life form. And this is something that we want to mimic in our culture media so that we can give it the best possible situation to grow on. So culture media is useful for the growth of the organisms and providing nutrition as in a natural environment. We should be able to understand the characteristics and properties of organisms. And then finally, these isolated organisms should be able to give us a biological product. Mind you, screening exercises are very cost intensive. And therefore, if you do not have a product in mind, you just cannot get along. It is simple, but the utilities or the consumables that go into such an exercise are intensive, and that is exactly what you need to be careful about. So what are the requirements of a microbe? The source of energy. We may have two different forms of microorganisms. One are chemotrophs, which require chemical energy, and one may be phototrophs. So if you're working in algae or you're working in any of the forms of life which are phototrophic, then they need radiant energy. So just providing them with sugar or just providing them with a chemical compound, these organisms will not grow. Second is the source of electrons. So either organic compounds can be sources of electrons, or lithotrophs, that is reduced inorganic compounds, are needed for the growth. Then you have the carbon source. Heterotrophs, which can grow on organic compounds, or you need if you have autotrophs, that is, you need to supply carbon dioxide as an energy source, a nitrogen source. Can it survive on atmospheric nitrogen? Does it need inorganic nitrates, nitrites, ammonium salts, or does it have a requirement of organic amino acids? Oxygen and sulfur sources are also important. Can it take oxygen from the water, or do you need to provide bubbling oxygen in the form of bubbles, pure oxygen? Is air okay? Sulfur components, do you need organic, inorganic, elemental sulfur? Phosphate component in the form of phosphate or phosphorus? What is it that you require? Trace elements are also needed. Now, trace elements usually are there as contaminants in the culture medium, but many a times this is not sufficient for growth. So, potassium, magnesium, calcium, ferrous, zinc, copper, all these ions need to be checked in quantities which are needed. Some organisms may also need vitamins and vitamin like compounds which function as coenzymes or building blocks for the coenzyme. Here, literature gives you an idea based on your end product whether this is needed or not. And definitely water, because this is the suitable solvent for almost all the metabolic reactions. And due to its specific heat, it also tolerates a little bit of sudden temperature changes. So based on your nutritional requirement, whether you need to isolate an autotroph or a heterotroph or chemotroph or an obligate parasite, you need to pick up our media composition in such a manner that you will selectively have these organisms grow. So knowing what you need in the end is also important because that will help you to define what you have, what you would give the organism as a food material or media for its specific growth. Now, when we are looking at media design parameters, we basically have to focus on which stage of microbial culture development are you? Are you trying to have an organism grown from a soil or a natural environment? Or are you looking at you have already isolated your organism, but it is with closely related other organisms? Should you look at selective growing, or should you look at just one that simple organism isolated further? Or you are looking at characterization of property. As I mentioned, each of these stages will have different nutritional requirements, and therefore media designing is important. Though the basic rules are the same, that you have different source of carbon, nitrogen, and energy, these rules are the same, but for each of the stages that you have are in media designing for that particular stage is important. I'll give you an example, and I want participants to come up with their thought processes. So I have this organism which is growing in marshy salt water. Right? It is basically this in the muck of that saltwater creek. I have got a sample of that saltwater soil. What will I do next? Can I have responses? Can participants unmute themselves and tell me what would you want to do? Anyone? I have 20 participants other than me. Anyone can tell me what you would do? My starting point is I have soil from a marsh, saltwater marshy region, and my final goal is to isolate a fast growing organism from this saltwater system. I'm okay if it is salt tolerant. How would you design? Yes, you will prepare media Saxena, but how would you go about the whole process? What do you think would be needed in the enrichment process? Okay. Nobody wants to say anything. So if I have to isolate from the soil will have a lot of soil particles, and in that the concentration of the organism of interest may not be high. So the first thing that we will do is we will bring that soil to the lab and allow the organisms in that to thrive by giving it an enrichment. So what we do essentially in this case is we give a good source of carbon, nitrogen, energy, and also of the salt in the area, because if it is a salty marsh, the organisms living there are tolerant to the 2 or the 5% salt that is there in the environment. So you give enough of carbon and nitrogen sources. So you give it glucose and you give a good peptone source along with the salt. You design this enrichment media where the organisms can thrive. Now, how do you measure whether the organisms have grown properly? By doing a staining of the supernatant or doing a absorbance, or you say the OD, optical density measurement of the supernatant, which can be an indicator. But we usually do a gram staining to see whether the organisms have grown or not, or if the media is helping them to grow. Okay. Now, once we have done this and we have grown the organisms, we have given a chance for the microbes that are growing there in that environment to thrive based on the media we have given. We pick up a sample of that enriched media and then put it further onto a system which will selectively allow growth only of the organisms that we are expecting to find or the properties that we are expecting. So in the first set, if you have given glucose as a primary source of carbon, and you want organisms which can selectively utilize only xylose or arabinose, which are other forms of carbon, or you want it to digest starch and grow on that. So in the selective medium, we will introduce that component and pick up organisms which can digest starch and then grow very effectively there. So even during this selection, we may have 10 to 15 organisms which are growing under those criteria. So the next level of criteria was what we wanted, a fast grower. So in the isolation step, we put up certain restrictions wherein we see only fast growers growing up. And once you have isolated this, finally, you look at the property that you are looking at. Because in selective media, we defined that we want starch degraders. Then we look at, after isolation, we look at have organisms which can produce amylase, which will be effective in starch degradation, and then the entire process can go. So these are the holistic parameters in which we design our different steps so that the selection can be much efficient. So let's quickly look at the basic composition of a medium. Water, carbon source, nitrogen source, energy source. Sometimes we use just the carbon source as the energy source. In other cases, when we know that, okay, there is a specific requirement for energy, then we add small molecules or metal ions or any sources that are essential. Mineral salts and then special growth factors like vitamins are also. When we are designing the medium, we take into consideration the physical requirements, the chemical requirements, whether we want anaerobic growth in the system or we are okay with just the normal aerobic. Is our microorganism microaerophilic? All these parameters can be introduced into the culture medium when we are defining them by use of different media and components. And finally, we look at what is the bacterial population that we have in there. So media can be of two types, can be classified based on two types. One is physical state, you have liquid, solids, semi-liquids, and semi-solids and liquid in one of in the physical type. And based on ingredients, you can have synthetic or undefined medium and complex or undefined medium and also specialized medium. So when we are defining the definition of defined medium, this would be wherein we know all the quantities of the ingredients. A defined source of carbon and nitrogen usually. Glucose and glycerol are usually used as carbon sources, and ammonium salts and nitrates are used as inorganic nitrogen sources. And then you look at undefined source, undefined group medium. Here we have complex ingredients. These consist of a mixture of many chemical species, which you may have in unknown proportions. And then you have yeast extract, casein hydrolysate, or maybe feather hydrolysate, hair hydrolysate. Whatever you can think of can be used in the undefined category. So we have an example of both of them. When you have a defined medium, I have every component that is put there, which is defined. Glucose, NH4, H2PO4, NaCl, magnesium chloride, K2HPO4, water, every component is defined. On the contrary, when we are looking at complex medium, we do not have that kind of differentiation. Peptone. A peptone can be a nitrogen source or a glucose. It will have some quantity of glucose also in it. It will have all these salts in there. Beef extract is also protein hydrolysate and amino acids. But in addition to that, you will have metal ions and other contaminants also. So here we are not defining the medium. Agar is basically used for gelling making a solid matrix, solid or semi-solid matrix. So these three components that are added in there are not defined through their chemical nature. They are just crude material that are taken in, and we may have estimations of nitrogen content, carbon content. We could take that as a parameter. The specific medium that are designed here, as I mentioned earlier, can be enrichment medium. This may be similar to selective medium, but here the focus is that your microorganisms in a given sample need to be amplified multiple times so that you are basically bringing them to a detectable level. But we should keep in mind that we are not simulating the entire bacterial population, but just the microorganism that we are interested in. Selective medium are, in this kind of medium, what we do is we introduce certain agents wherein we want to suppress the unwanted microorganisms and grow the desired organisms. In differential media, we would usually have this as a solid medium, but here the colonies will be differentiated from one another based on the color. So for molecular biology studies, we use X-gal plates wherein the transformed colonies look nice and blue, whereas the untransformed colonies look white. So this becomes the media is selectively picking up the ones that are transformed. So this kind of media is also essential in our studies. Then we have transport medium. Basically, transport media are solutions of buffers, carbohydrates, peptones, wherein the viability of the bacteria is preserved during transportation, but we do not allow their multiplication. And lastly, we have anaerobic media, where growth of obligate anaerobes is possible. So for every specific organism that you want to isolate, so if you're looking at bacterial isolation, the parameters are different. Actinomycete isolations, the media is different. For fungi and yeast isolations, the media is different. And all these media designing need to be paid attention to during your So this is one small work that we had done way back in 2006, 15, 16, where we had taken samples of organisms from rotting seaweeds and tried to grow them. So from the rotting seaweeds, we had enriched and plated out around 367 from fresh seaweed samples and 400 from the rotting seaweed samples. And all these samples were then put up in selective media where individual enzyme properties were characterized. So the entire 800 colonies went on to plates in this manner, where individual colonies which gave up, which showed the property or showed the enzyme behavior were picked up. So here first level was an enrichment kind of a activity, enrichment and isolation that we did. And then the second level, basically we used different selection medium to pick up the organisms which exclusively show those properties. And we were able to isolate around 29 distinct cellulose and bulwan saccharifying organisms. So this is the part wherein we identify our organism, and then this particular organism needs to go to the next level where we need to grow it for the product that we are using. Now here there are two fermentation media that we need to look at. One is the growth medium, where the organism can grow very healthily, and this provides the raw material for further fermentation. So in this grow material, we have low amounts of nutrients, but we are expecting the cells to grow faster. And once the cells are grown faster, then we transfer them to the fermentation medium so that they create the final product that we are interested in. For example, growth of the yeast needs just 1% carbon. But when we are fermenting alcohol or we are fermenting glucose to produce alcohol, we usually use around 10 to 13 or rather 15% of glucose to allow the alcohol fermentation. Right? So the growth medium is just to support growing, whereas the fermentation medium is basically to... Yes? Can you see the screen?
Yes, Professor.
Yeah. But the fermentation of alcohol, we need higher concentrations of glucose because we are interested in the final product alcohol. We're no longer interested in the growth, but we want the product. So wherever we are looking at growth medium, we in a systematic manner need to increase our cell mass in such a manner that this cell becomes the catalyst for the further conversion in the fermenter. So microorganisms grow on or in medium which satisfies their nutritional needs. So this growth medium for production scale just satisfies their nutritional requirements. And we pick up this so that it is a favorable medium which is allowing it to grow by combining water, carbon, nitrogen, and other supplements. So the criteria for picking this up is that the cell growth or the growth curve of the cells should be optimum, and the growth, we should be able to calculate the microbial growth rate in these medium, and it should be at the highest case possible. So when we are designing growth medium, our requirements are that we should get maximum yield of the cells, no side products, consistent production should be possible, and this should be a economic methodology of production. However, when we go to the production phase, we are looking at what is the final product that we are, and which carbon, nitrogen, and energy source is going to give us this product in the highest yield that is possible. With yield, we say we calculate based on the carbon input or the nitrogen input, which is your starting substrate, and how much product can you make out of that. In many cases, we do allow growth also in this case, which is called a classical fermentation. But when we are referring to transformations, we have fully grown cells which are inoculated in this system. so that we have just the product formation and minimum byproducts that are formed. There are multiple programs and there are multiple methodologies of going through a systematic approach for fermentation designing, where you have criteria that are used and any changes in the criteria are reflected by doing things. So we have a database of, this is a paper where strategies of fermentation media optimization has been reviewed. And one of the screening methodologies or the flow sheet methodologies that they have given is based on these parameters, where they are looking at C by N inducer ratios and identifying the C by N ratio sources which you can have, that is carbon by nitrogen ratios, are very crucial for the growth or the transformation phases. And they are different. When you are growing the cells, the nitrogen C by N requirement is different. And when you're fermenting, the C by N ratios are different. So in both of these cases, how you should plan the system so that you have optimum growth of the system is absolutely essential. So therefore, this input becomes a significant introduction into the production medium. And then these optimizations, we use a lot of statistical design and optimization at the shake flask level. So complete design of experiments can be done for coming to the final evaluation of results at the shake flask level. And once in this shake flask level, we have identified that X, Y, Z are the parameters controlling. The similar controlling factors are into the next level of optimization, wherein you are doing on a bioreactor and optimizing the media, whether it works at the bioreactor. So again, the requirements of the fermentation media is that you should have full active growing cells. They should be allowed luxurious metabolism. They may not be growing, but they should be allowed to do the metabolic activity that we are interested in, so that we get the final product. In your fermentation, they become hyperactive because you have high quantities of nutrients, and the excess nutrients, the consumption of nutrients happens, and we get degradation of the fermentation media also. This is important because waste effluents that are secreted are our final desired output product. But we do not want that there are other byproducts in the system. Therefore, optimal control of what you are feeding or the media that you are feeding it has to be optimized so that the waste effluents are met. Now, again, in this category, we have two types, synthetic medium and crude medium. Synthetic medium, as the name suggests, everything here is controlled. You have a very smart design that goes into the synthetic medium, and it is able to control the product growth and the yield of the product in a lab environment. Even if we do a smart design of media, we can actually trace the metabolic pathway also in many of the cases. On the contrary, crude media are basically undefined sources of ingredients, but we pick these up because it gives the high yield of product, and you are not needed to add other small ingredients which increase the cost of the final money. So it's also important that we have to screen the crude media that it doesn't contain toxic materials for the final. There are a lot of advantages and disadvantages of both the systems. The advantages of the synthetic product are you have no foam formation, chances of contamination are very low. So it's a well-designed medium where you know amino acids that are going in. Everything is properly defined, and therefore product recovery becomes very easy in these kind of systems. Whereas on the contrary, the major disadvantage is the cost. This becomes very expensive to be done at the industrial level, and therefore synthetic medium is usually used only at the lab scale. Crude media, on the contrary, are largely composed of materials from the nature, and we pick them up because of the low cost which then influences the fermentation process. And these may be varied in composition. You may have batch to batch variation. Seasonal availability is also a problem in this. But looking at the cost economics, we finally pick up this. The selection criteria or the needs of the microorganisms are then same. So depending on what is needed, we pick up inorganic nutrients, carbon sources, nitrogen sources, growth promoters, precursors, and buffers, which can be used in these to design a perfect media for optimization. Looking at when we come to the optimization per se, you can have closed-end optimization or you can have open-ended optimization. In closed-end optimization, we take a fixed number of components or component parameters that can be used for the optimization. This is a simple strategy, and many components that are beneficial may not be sometimes considered in this because you have a limited set of experiments that are focusing only on the major parameters that you think are important. For an open-end parameter, it is highly labor-intensive, but it makes no assumptions that which component is important. It keeps on, in this process, we keep on optimizing till we come to the final. All products, all components and parameters are taken into criteria. So the optimization technique basically has traditional one factor at a time, or it can involve statistical and mathematical. So one factor at a time, you have removal, supplementation, replacement as the core aspect of the one factor, and you take into consideration your different amino acids and keep replacing them. This is a traditional way of doing it. However, in advanced methodologies, you see that you use statistical and mathematical tools, wherein screening of significant components is done by the Dagaji or the PDB method, whereas you also have parameters of RSM and artificial neural networks, genetic algorithms, fuzzy logic. All these are also used for optimization. So a whole lot of methods are currently being used where borrowing, biological mimicry, one factor at a time, factorial design, Plackett and Berman design, multiple mathematical and statistical parameters are being used. And if you see, there is a whole spread of papers that are coming out, literature that is coming out, which caters to each of these methodologies for application for optimization. And that brings us to the point where people are trying to do automation in media design. So as we all know, we have only 1%, 0.1 to 1% of microbial organisms that are very successfully isolated. So therefore, Richard et al. And team, they developed a computational modeling database, which helps our culturing efforts. And here they have used metabolic reconstructions on organism-specific sequences and biochemical knowledge to design the media. And MediaDB is the website which has this. So here, after you input your requirements of the genus, the organism that you are interested in, and the biomass content that you want, you could actually calculate the growth data and the media components that are responsible for giving you a particular data calculation. Now, using this database, what they have done is they have filled up all the literature that is available for experimentally verified media formulations, and you have chemically defined media so that every compound is linked to the metabolic network. And once you have such linkages, then this is further linked to compound identifiers, and you can cross-reference with other sources. And this focuses on organisms with existing in silico models, because that is where prediction can be done more appropriately. And once you have used this, it serves as a set of organism-specific media conditions so that you can have products that are designed as per the metabolic pathways to give rise to media formulations that work at all point of time. So it also includes species with fully sequenced genomes to ensure media formulations can be tied back to the genomic database. People are also using a whole lot of artificial intelligence in media optimization, wherein the entire focus is to have towards the metabolite yield. So optimization of the production medium is done towards the metabolic yield, and and techniques that are being used are one factor at a time, and modern statistical methodologies like genetic algorithm and artificial neural networks are also being used. The problems associated here are the range of experiments that you have to do is very huge, and from the isolation of the organism to the final medium that you are using for growth and fermentation, this may not be the same. And at each point, we need to do media optimization for the growth of the particular concept. And the other aspect is many a times what happens is the shake flask data and the fermentation data do not match with each other. What you get best on a particular medium in the shake flask may not work in the fermentation. And it is widely assumed that the best medium that as per standard practices, we think that whatever comes best in the shake flask will also work in the fermenter. But this is not the case because aeration, the reducing potential in multiple cases, then the mass transfer of the substrate to the organism, all these parameters would be very different in the fermenter. And therefore, rigorous studies need to be done at the fermentations. So when we are designing the fermentation medium, this is always a never-ending process. And with a whole lot of internal control mechanisms, which we don't understand, we usually do optimization studies as a black box. But what most people do in the area is to focus on the components which will give you maximum yield so that your designing has a perspective. You have to have an end goal so that your designing assumes a certain perspective. The future in this direction, the media formulations basically are evolving because we are able to screen by carbon, nitrogen, and different combinations for growth. So in the earlier century, people were not looking at biomass or lignocellulosic biomass as an option for microbial growth. We were not looking at rice bran or multiple of these agri-based waste industry to come back as microbial growing resources. Right? So today we are looking at these various carbon and nitrogen sources to pump in the microbial growth. And because of this, you have a lot of options in the media formulation that can come in. And there are still certain possibilities because microorganisms have limitations at the genetic level for production of a metabolic. So this search is always needed, but we have to base it on more precision and optimization. And sometimes you have organisms which can utilize cheap raw materials, and through mutation, we should be able to do this. So in conclusion, the designing of a fermentation medium can be a never-ending task, and every optimization technique has its own advantages and disadvantages. The Blacklit-Berman design and component replacing can be a good methodology to go through the screening methods, but recently neural networks, fuzzy logics, and all these AI-based tools that are coming in will be significantly allow us to define our media properly with proper resources. So this is the place where I come from. We are basically multitasking and a multidisciplinary center. And thanks everyone for your time.
Thank you so much, Professor. The session is open for discussion. If you have any questions, you can ask Professor. Any questions, students? You can also use the chat box to ask questions if you like. Okay.
No, the system, according to time, you will not be able to change the real variable. You will have to have a set of parameters where you define, and then we can use variables. So why we use two variables, real and dummy? Because these what do you say, the parameter, I mean, that's what in Blackett Berman it becomes a little bit stringent, where you have either one option that is functioning very well and one that is zero. But what essentially we look at is the reduced number of experiments that you have to do to quickly come to a solution. If you, that's what that is where the difference between closed end and open ended optimization comes in. So if you are looking at open-ended, you will keep on doing the experiments till you come to a final conclusion. And in that, you can have multiple variables that come in. But if you're looking at a closed-ended system, your multiple variables are not there. And you have a fixed number of variables and a fixed number of parameters that you are working with. And that allows you to narrow down only till a certain point. A detailed optimization is not possible. But it's a race against time versus your effort. Multiple times when we have the screening experiments, we are only given 6 months for optimizing the media that you want to use. Many a times this falls short, and the number of experiments you have to do is much more, and the optimization to come to a final conclusion.
Any more questions? So if no more questions, we will. I would like to thank Professor Annamma for joining us today. Thank you so much, Professor, for giving us your valuable time and sharing with us. Thank you so much.
Thanks so much. Thank you. Thanks everyone for attending. Thank you for the opportunity. Yeah. Bye.
That's all for the day, students. We'll get back tomorrow with another set of lectures. Take care. Thank you. Thank you all.